All right, let's get into a really cool scientific detective story. Today, we're going to untangle the incredibly confusing evolutionary history of a huge family of proteins. And we're going to see how a totally new way of looking at the evidence finally cracked the case. So, imagine this. You're trying to trace your family history, but every single record you find tells you a completely different story. Some say your great-grandparents came from one continent, others say a totally different one. It's a mess, right? Well, that is the exact problem scientists were facing with this group of proteins, and it created a major biological mystery. And our mystery kicks off right here with this fascinating and, honestly, enormous family of proteins called the serpins. So what in the world are serpins? Well, they're this massive superfamily of proteins that do all sorts of critical jobs in the body. I mean, some of them act like molecular police, stopping other enzymes from going haywire. Others are kind of like delivery trucks, carrying hormones exactly where they need to go. And they're everywhere. Animals, plants, even viruses, which makes figuring out their family history super important. Now, to map out that kind of history, scientists build what's called a phylogeny. It's really just a fancy word for a family tree, showing who's related to whom and tracing their lineage all the way back to common ancestors. And getting that tree right, it's absolutely fundamental to understanding how evolution works. And this isn't just some small academic puzzle. The stakes are actually pretty high. As this quote points out, getting phylogenies wrong can create these massive contradictions in how we understand the entire history of life. When your molecular data says one thing and the fossil record says another, you've got a serious problem on your hands. Which brings us right to the heart of our case. For years, every time scientists tried to build the Serpin family tree using the standard methods, the results were just a complete mess. Different approaches gave you completely different trees. It was this tangled, contradictory web of relationships. The usual tools just weren't cutting it. So to solve the mystery, researchers needed a new angle. They decided to look for clues not just in the final protein, but in the original blueprint itself, the very structure of the genes in our DNA. And to get this clue, you just need to know how a gene is put together. Think of it like a recipe. You've got the exons, those are the actual instructions, the ingredients for building the protein, and then you have introns, which are these non-coding bits that get snipped out before the protein is actually made. So exons are the key parts, introns are just the spaces in between. Okay, take a look at this. This slide lays out the genetic blueprints for different serpins. The black bars, those are the exons, the important instructions, and the thin lines are the introns. Just by looking, you can literally see that these groups have totally different architectural patterns. The design for one is completely distinct from the next, and so on. And this was the first huge breakthrough. By focusing on the gene's physical structure, its unique pattern of exons and introns, the researchers could finally sort that chaotic mess of serpins into 6 distinct, really well-defined families. It was like sorting a jumbled pile of architectural blueprints into 6 different styles of buildings. But, good detectives always need more evidence, right? So for their second clue, they looked for a kind of secret code hidden inside the proteins themselves. Something they called amino acid fingerprints. So a diagnostic site is a very specific spot in the long chain of a protein where every single member of one family has a unique set of amino acids that no other family has. It's a chemical signature, a fingerprint that basically says, if you have this specific marker, you belong to this family, period. And this table here shows it perfectly. Just look at position 110. Every single member of the group 2 family has one specific amino acid, Y, right there. But in group 1, you find a bunch of different ones, but you never see Y. It's an absolutely unmistakable marker that powerfully confirmed the groups they'd already found by looking at the gene blueprints. Okay, so now they had 2 powerful and completely independent lines of evidence. One from the gene's architecture and one from the protein's fingerprint. The investigators were finally ready to put all the clues together and solve this case once and for all. So their investigative method was this brilliant 3-step process. First, group the genes by their physical structure, the blueprints. Second, confirm those groups using the amino acid fingerprints. And third, for just rock-solid proof, they looked for one last clue, these rare genetic events, like insertions or deletions, which are really strong evolutionary signposts. And here it is, the big aha moment. When they combined all 3 clues, those 6 messy families just collapsed. They fell neatly into 2 major supergroups, or classes. Class I is defined by having a specific intron at one position. And Class II, it's defined by not having that intron, but having other unique features. All that chaos was finally resolving into a clear, simple structure. This was the ultimate prize. By combining these different independent clues, the researchers created the very first reliable, internally consistent, conflict-free evolutionary tree for vertebrate serpins. The tangled web had finally been straightened out. But, solving the puzzle wasn't even the end of the story. Because this new, correct family tree, well, it allowed them to tell a whole new story about how these complex genes actually evolved in the first place. So this stable tree let them answer one last huge question. How did these genes get so different? Was the original ancestral gene super complex, with lots of introns that were then lost over time? Or was the ancestor's gene simple, and it got more complex as new introns were gained over millions of years? The data gave a stunningly clear answer. The probability of that intron loss model creating the patterns we see today was calculated to be less than 0.9%. It's statistically almost impossible. The idea that all these different groups just happened to lose introns in such a specific, coordinated way was just unbelievably unlikely. And this led the researchers to a really powerful conclusion. They strongly favored a model where evolution was dominated by intron gain. It's just far more likely that the ancestral Serpin genes started simple and got more complex over time as new introns were added, creating the diverse blueprints we see today. So why does this one case matter so much? Because this isn't just a story about serpents. This is a proof of concept. It shows that by combining clues from totally different biological features, a gene's architecture, a protein's sequence, you can build a much more robust, much more reliable picture of evolution. It's a powerful new toolkit for other scientists to use. And that leaves us with a final thought. This was a case where the standard approach just failed, and the solution came from thinking creatively and looking for evidence in places no one had thought to look before. It really makes you wonder what other tangled family trees in biology, or maybe even beyond, are just waiting to be solved by someone willing to look for a whole new kind of clue.